Hello, welcome back to the second part of the video where we were discussing about the process of transcription. So in the first part, we have discussed about how the process of transcription has been taking place in the prokaryotes. And what is the general process of transcription and what do you mean by transcription we have already discussed in the first part of the video. Okay, that is prokaryotes. If you have not watched the prokaryotes, I request you to please watch transcription in prokaryotes so that you can understand better the comparative study between the transcription and the in eukaryotes as well as in the prokaryote. In this part of the video, we are going to discuss about the transcription in eukaryotes. So let's begin. Before that, if you remember, in case of prokaryote, the transcription process starts directly. That is, there is no need of regulation, no need of certain extra things to be done. Why? Because the DNA is nearly circular in nature and the DNA is very simple in case of prokaryote, not much complicated. Whereas when it comes to eukaryotes, the DNA is linear DNA as well as the DNA consists of histone proteins and certain foldings are present in the DNA, which makes the structure of DNA really complex in case of eukaryote. For that purpose, the regulation process must be done. What is regulation? Regulation means that is certain modification is needed to be done before the process of transcription is initiated. That is known as regulation. Then the transcription process like the initiation, elongation and termination takes place, but this doesn't end here. After transcription also, post-transcription modification is done in case of eukaryote. Why so? You must be wondering why so? Because in case of the eukaryote, for the translation process, the that is the mRNA need to be transported to the cytoplasm. Okay, for that purpose certain modification is needed to be done so that it does not get degraded in the cytoplasm. Okay, this is very important to be noted down. So the process of transcription, you can generally say in eukaryote, is divided into three parts. One is regulation, second is transcription process, which belongs to initiation, elongation and termination, and next comes to the post transcription modification. In this part of the video, we will be discussing mainly about the first two part, that is the regulation part. Okay, next we will be discussing about the transcription process. Post modification includes the splicing, capping and polyadenylation process, which we will be discussing in the later part of the video. Okay, so for this part of the video, this much is enough. Okay, next coming to the first part of the process, that is the regulation part. Before that, you know that for the transcription process to be conducted, very important enzymes is needed for the process of transcription. What is that? That is known as the RNA polymerase. Okay, RNA polymerase is a very important enzyme which is necessary for the process of transcription to be completed. And the transcription in case of that is eukaryote, there are three types of RNA polymerase, that is RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. Okay, so this table is very much important regarding exam point of view, because it is asked generally that which type of RNA polymerase synthesize which type of RNA. Okay, like RNA polymerase 1 will synthesize rRNA as well as 5S rRNA and 28, 23S rRNA. All the subunits of rRNA will be synthesized by the RNA polymerase 1. Okay. Next, coming to the RNA polymerase 2. RNA polymerase 2 will synthesize mRNA. Along with that, miRNA, snRNA. What are miRNA, siRNA? We will be discussing when we will be studying post transcription processes. We will be dealing with that later on. And RNA polymerase 3 mainly deals with the synthesis of tRNA. So this is the transcript, or you can say these three are synthesized individually by RNA polymerase 1, 2 and 3. Okay. Next, coming to the site of action, RNA polymerase 1 is mainly located on the nucleolus, whereas RNA 2 and 3 are located on the nucleoplasm. Next, coming to the promoter region, the RNA polymerase 1 and 2 are located in the upstream, whereas here is a little mistake. The RNA polymerase 2 is located on the downstream. I have already told you what is upstream and downstream. If this is the initiation site, towards the initiation site is known as the downstream. Opposite to that of the initiation site is known as the upstream. Okay, this is known as downstream, this is upstream. This is very important to know that which is downstream, which is upstream, because this is the main confusion area. Okay. Next, coming to the alpha emitting activity. Alpha emitting activity generally means the bacterial activity. Okay, how much the bacteria mainly will affect this RNA polymerase. Like they have RNA polymerase 1 will have no activity to that of the bacterial. RNA polymerase 2 is highly sensitive to this activity, and RNA polymerase 3 shows medium sensitivity. Okay. This is all about a brief description about the RNA polymerase. Moving on to the regulation part. As you know that the DNA consists of chromatin, okay, that is the chromatin structure, you know, that is thread-like structure. So for the chromatin needed to be regulated. How the regulation is done? First, the gene activator binds to the chromatin region. Okay. Then what will happen? The chromatin remodeling complex will come and bind, and the chromatin remodeling will take place. Then histone modification enzyme will be added, which will modify the histone covalent bond, along with that other activator protein and additional activator protein along with the general transcription factor will result in the complete modification of the DNA and starts the transcription process. So what is really happening? Let's see. So this is the chromosome. Generally the chromosome is present in this folded manner. Okay, this is highly folded, that is folded 30 nm fiber. You can see highly folded looped DNA is present. So to release this loop structure so that the process of transcription takes place, histone modification, chromatin remodeling as well as RNA polymerase, these three will take part to make the genes in this. You have seen this region, that is it was looped and very constricted. Now they will open this region so that the process of transcription can take place. So this is known as the regulation of the chromatin. Okay. Next, moving to the two most important term, that is HAT and HDAC. Okay, what is HAT and what is HDAC? Okay, if you understand that the chromosome consists of the, that is the DNA are compact structure, they consist of the histone protein. Okay, so HAT is known as histone acetyl transferase. That is what it does, it adds acylated histone on the N terminal along with that of the activator and co-activator, which makes the chromatin activate. If you see, this is a chromatin. Okay, this is a chromatin and these are histone. Okay, so here what will happen, this black color structure you are seeing, this is known as the HAT protein. Okay, or you can, or HAT enzyme. Basically acetyl transferase means what? Transferase means it will do the work of transfer, acetyl group transfer it will do. Okay, so normally if you understand in simple word, acetylation will lead to the transcription starting point, or it will on the transcription. Okay, acetylation means transcription will be on and transcription will take place. Okay, so acetylated histone means that is they are active chromatin, and non-acetylated means inactive chromatin, and the process of transcription does not take place. So what does the HAT protein do? HAT enzyme along with that of the activator and co-activator will bind to that of the chromatin structure. Okay, as a result of which the acetylation will take place. As a result, the transcription process will be on. Just opposite activity is done by HDAC enzyme or protein, you can say. HDAC is known as histone deacetylation complex. It will bind with that of the co-repressor as well as repressor and will stop and cause the deacetylation, as a result of which the chromatin structure, you can see here the chromatin structure is again folded back and form the inactive chromatin. Here you can see the chromatin is stretched out so that the process of transcription gets easily done, but HDAC what is done, it mainly makes the chromatin inactive and does not allow the process of transcription to be done. Okay. And one more thing which prevents the acetylation is the methylation process. Okay. Indirectly you can say if the methylation process takes place, then the transcription process is off. Okay. So the acetylation will lead to the transcription on, methylation will lead to transcription off. Okay. This is very important to be understood, these two proteins. And there are two regulator proteins which are present, which mainly enhances the activity of the transcription. What do you mean by enhance? Enhances means it will increase the activity. Regulated protein, one is enhancer, another one is silencer. What is the activity of enhancer? It will increase the activity of the gene promoter region. As you know, the promoter region is important for the binding of the RNA polymerase so that the process of transcription takes place smoothly. To increase that one activity, the enhancer is mainly done. Okay, the enhancer is distance free, orientation free and position free. So wherever you will place the enhancer, it will help the promoter region to enhance its activity. So it does not depend on distance, orientation or position. Wherever you will place, it will act. Okay. The opposite activity is done by the silencer. It will replace the gene activity. It will stop the gene activity. So these are the two regulatory genes that will help in the process of regulation of transcription. So this is all about the first part of the transcription, that is the regulation process. Hope you are clear with it. Moving to the next part is the actual process of transcription, which is known as initiation, then is elongation, and finally the termination process. How does it take place? We will discuss. First, what happens? A pre-initiation complex will be formed. Okay, a pre-initiation complex will be formed along with that of the general factor. What are the general factor? Will combine with the RNA polymerase 2 to form the pre-initiation factor. What are the general factor? Like the TF2A, TF2B, 2D, 2E, F and H. Each one of them has certain specific role which plays the important role in the process of initiation process. Not only initiation but also in the activity, further activity. Before that, let us focus first upon the promoter region. As in case of the prokaryote, you have seen that the promoter region consists of the -35 sequence, Tata box and start sequence in case of prokaryote, but in case of eukaryote, that are four type of structures are present. One is BRE sequence. Okay. Second is the Tata box. BRE sequence is located in the -35 region, and Tata box at -30 region, and transcription start point INR is present right here, and DPE, that is downstream promoting factor, is present in the +30 region. So this is the promoter region. Okay. Next, what are the role of each of these general transcription factors? We will be discussing. TF2B has the that is the role of BRE recognition element. What is BRE? What does the role of BRE mainly do? BRE is responsible for binding of the RNA polymerase here, and then it will slide against this and reach the initiation start point, and the process of transcription will take place. So the TF2B, this is very important table. Which general factor is responsible for what type of element activity? So TF2B is responsible for BRE recognition. TF2D, that is TBP, that is Tata box protein. Okay, this is responsible for the Tata binding protein. Okay, Tata binding protein is responsible for recognizing the Tata box. TF2D is responsible for binding to the INR, that is the initiation site, and TF2D is responsible for binding to the DPE. So the mainly you can see that TF2D is responsible for binding to Tata, INR and DPE. Another interesting point is that the TF2D contain Tata binding protein. Okay, Tata binding protein will bind to the minor groove of the Tata box. If you see here, this is the promoter region, okay. BRE sequence will be present, there is Tata box, this is start signal, and this is DPE will be present. So what will happen, the TF2D has the, you can see this is TF2D, TF2D has the TBP, that is Tata binding protein. It will come and bind here, okay. TF2B, 2D will bind here and it will recognize the BRE sequence, okay. Recognize the BRE sequence and accurately and positions the RNA polymerase at the start site of the transcription. So what will happen? Once the TBP binds to that of the Tata box, it will first bind to that of the, that is Tata box region. Then what will happen? It will recognize and then what will do? It will call the TF2B, another factor it will call, and this TF2B, what is the role of TF2B? You have seen here, that is BRE recognition element. So next what will happen, that if you see here, the TF2D, sorry 2B has bind here, and then what will do? The TF2B will recognize the BRE element and will accurately make the RNA polymerase bind here, and the process of transcription will be initiated. All this process we will be discussing now in detail. Let's start. First what happened, this is the promoter sequence, this is the BRE sequence, this is the Tata box sequence, initiation point and then start point, and the DPE sequence that I have shown you, okay. So what happened, first what happened, the sequence should be remembered, because the sequence is mainly asked in exam, or you can be asked in the sequence wise question, or match the following, which comes first on that it will be asked, okay. So first which transcription factor will come? TF2D along with the Tata box binding protein, it will come and bind to the Tata region, okay. Immediately after the binding of the TF2D, it will call, first what it will call? It will call the TF2A and TF2B. So TF2A and B will work simultaneously, okay. You can see here, this is the sequence I have given. First TF2D binds with the Tata box, then what will happen? TF2A and 2B will come simultaneously. Then what will happen? TF2F along with that of the RNA polymerase 2 will come and bind to this region, okay. TF2F and polymerase 2, that is, you can say that TF2F will bring the RNA polymerase 2 and bind to this region. Then gradually it will bring the TF2E and H. So the sequence, if you remember, first which will come? First D will come along with that of the TBP protein, and then A and B will come and bind with it, okay. What is the role of B? B is responsible for the binding to the BRE sequence and recognition, so that the proper binding of the RNA polymerase takes place. Then what will happen? F will bind, okay. F will bind, F will bring the RNA polymerase along with it, so that it will be binded properly. Then after F, E will come, okay. After that E will come, then H will come. So this is the sequence of the binding of the transcription factor, which is very important because the sequence is mainly asked. Okay, this is the role of each of them. What is the role of TF2D? TF2D has Tata binding protein, and TAF, Tata binding protein, will recognize the Tata box, as I have already told you, and TAF will recognize the other sequences present in the promoter region. Next, what is the role of TF2B? TF2B will recognize the BRE sequence, which help the RNA polymerase to bind to the start site. I have already told you. Then TF2F will come and bind with that of the, will bring that RNA polymerase and will bind to it properly, and along with that it will attract E and H factor. E will attract, what is the function of E? It will attract the H factor, and H activity is mainly have two roles, that is helicase role as well as nuclease role, that is 3' to 5' as well as 5' to 3' hexanuclease activity is also done, as well as helicase activity is also done, that is it unwinds the DNA. It is very important factor, okay. it will unwind the DNA at the transcription start point and phosphorylates the serine 5 of the RNA polymerase CTD region. What is CTD region we will discussing now only, and release the RNA polymerase through the promoter. So first it will help in unwinding, then it will help in phosphorylation, and finally after the process of transcription is over, it mainly releases the RNA polymerase. So major activity is done by the TF2H. What is the role of each of them? I have mainly mentioned in this table, so do go through this table once for the better understanding. Next, coming to the RNA polymerase 2 is responsible for the process of transcription in eukaryote. So RNA polymerase, if this is an RNA polymerase has a sequence, that is a tail sequence, which is known as the CTD sequence, that is C-terminal domain sequence, which is the largest subunit. It is known as heptamer because it consists of 7 subunit, okay, 7 subunit, and this is responsible for the phosphorylated, it is phosphorylated at serine and threonine region. This CTD is responsible for the capping, 5' capping, splicing and polyadenylation process. Okay. So when we will discuss about the post transcription modification, we will be discussing this one in detail. For now you remember this much. Next, coming to the elongation, for the process of elongation, CDK9, that is the CDK9 is part of elongation complex, as well as 2 other factors like the PTEFB and eukaryotic TF2S will be responsible. Okay. TF2B mainly positive transcription elongation, that will help in the elongation process, and it is cyclin dependent kinase, okay, CDK9, that is cyclin dependent it is, and it will work in a slow process. Okay. Whereas eukaryotic TF2S is another elongation factor, it will overcome pausing. What is overcome pausing? That is, it will help in the smooth running of the RNA polymerase along the DNA strand. No pausing will be taking place and it is a fast process. Okay. Next, coming to the termination, as you have seen in case of prokaryotic termination, it was row dependent and guanine rich sequence will be present, but here no such extra additional protein is required. Thiamine residues will be present at the ends terminal. If this is the RNA, if it is the DNA structure, then at the end of the structure the thiamine residue will be present, which will lead to the formation of DNA RNA hybrid. What it will do? This is DNA and RNA is being synthesized here. So the thiamine residue will form self-complementary with that of each other, as a result of which DNA RNA hybrid will be formed and the transcription process will be terminated there. Okay. So this is all about the process of transcription in case of eukaryote. Half we have been done, that is we have only dealt with the regulation as well as the transcription process. Next part of the video will be regarding the post transcription modification, which is very important, that is splicing, polyadenylation, and next is the then is the capping. Okay, 3 processes are very important. Hope you have understood this part of the video. This is very important and informative. Share as much you can among your friends, because these are very interesting part as well as mainly important for exam point of view. If you are new to my channel, do subscribe, like and share as much you can. And this is for today. Thank you and have a nice day.